So today, it's a good thing that we'll be stepping a bit away from COVID 19. So I think one of the good things that this pandemic has done for us is that we now appreciate the role of science in developing our policies and how important it is in our daily lives. And some of the impacts or some of the research that is being done cannot be done overnight. So these are things that really need investment in terms of time and expertise, and I think the talk that we have today demonstrates just that. So I'm very excited to hear about this lecture. So today we're talking about evolutionary origins and spread of rice. Um, our speaker for today is a Silver Professor of Biology and the former Dean for Science at New York University. His research is on plant evolutionary genomics, and in recent years, he has focused on domestication and the evolution of crop species, including rice. Prior to joining the New York University faculty in 2006, he was the William Neal Reynolds Distinguished Professor of Genetics at North Carolina State University. He is the recipient of an Alfred Sloan Foundation Young Investigator Award and was named a Guggenheim Fellow and a Kavli Fellow and was awarded the 2015 Khalifa International Date Palm Award. In 2005, he was elected a Fellow of the American Association for the Advancement of Science, and he serves on the editorial board of various journals in plant biology and evolutionary genetics. And he is a trustee of the Alfred P. Sloan Foundation. Let us all welcome Professor Michael Purugganan.
So thank you, Kay. Thank you for the invitation. I had asked Kay a few days ago if, maybe it was not very appropriate for me to give a non-COVID talk. Can people hear me, by the way? Kay, yes? Okay. So the,
Yes, we can hear you well.
Okay, thank you. So I was wondering, but Kay said, maybe it's time to take a short break from COVID talks and to talk about something different. And so what I like to talk to you, and this is going to be hopefully a kind of a fun talk, in that at the end of the day, I think what you're going to gain is an appreciation for what I think is the history of rice. And hold on a second. Okay. And this is I don't know if people recognize this. This is what's called the New York kitchen sink pizza. It's a kitchen sink pizza because it's got everything on it. And you can buy it here in New York in some of the pizza places that you find in every other block in New York City. And when I teach a course at NYU called Bread, Wine, and Genes, which is a seminar course to study the evolution of domestication. And I usually start my course by bringing in this pizza. And then I challenge the students to count how many species are on this pizza and to identify them. And I'm not going to let you do that, but I'll tell you what they are. There are 10 plant species, there are 4 animal species, and there are 2 fungal species on this pizza that I can think of. I may be missing a few here and there. But what all of these species that we find, from the sheep's milk, cow, chicken, pork, oregano, wheat, olives, all of the species that you find here are essentially new species. These are species that have evolved the last 10, 12,000 years. These are what we call the domesticated species. And it's an area of study that I've become interested in really for the last 20 years. My interest in these species have accelerated. So let's first of all talk about what domestication is. Domestication is a special evolutionary process, and it comes across from a mutualism. What happens in domestication are there these 2 species: a domesticated species, which is one that has evolved to reproduce and grow under the care of another species, so that latter species derives benefits. So in the case of humans and let's say rice is a domesticated species, and we as humans are the ones that have prompted the domestication of rice about 9,000 years ago. And essentially rice sativa, is a species whose reproduction and growth is under our control. And that's true for every other domesticated species, whether it's plant or animal, is that the reproduction and growth of these species are under the control of humans. And the domestication takes place, this is an evolutionary process, because of this mutualism, when one species cultivates or tends another species. Now, humans are not the only ones that have prompted the domestication of species. Termites and ants also have developed their own agricultural system and have domesticated other species. But it's humans who have been the most prolific domesticators of all the species that we know of. So the way this has happened is because humans as a species, we've changed our own ecology about 12,000 years ago. So think about our ancestors before 12,000 years ago in the Pleistocene and prior to the Pleistocene. When our ancestors were trying to gather food, for example, they were gathering food. That was how they did it. They hunted animals and they gathered plants, berries, nuts, grass seeds. And that's how we fed ourselves. As an animal, that is how we got our nutritional resources. But starting about 12,000 years ago, and actually that date is now under debate, it might be earlier than that. Humans, Homo sapiens, this animal, changed its ecology. And it changed its ecology so that instead of gathering wild plants, it took the step of actually planting them and taking care of them and harvesting them from the plants that it tended in these gardens and then later on farms. It's this change in the behavioral ecology of humans from being a gatherer to being a cultivator that really caused the evolution of a whole series of species that became dependent on humans because humans were farming them and were getting their food benefit and sometimes fiber from them. So that's kind of the difference. The human change in ecology was the one that prompted the evolution of these different species. It's widely believed that this transition first occurred in an area called the Fertile Crescent. This is an area that goes from the Persian Gulf to the southern range of the Zagros Mountains in what was Iran, Iraq, and then southern Turkey and into the Levant. This area of the Fertile Crescent is where we believe the first domesticated plant and most animal species, many of the major animal species originally involved. And this is where we think agriculture evolved. And certainly this is important because with the rise of agriculture, we also see the rise of cities, the rise of organized social hierarchies and writing that occurred with the transition of humans from being hunter-gatherers to being agriculturalists. There's just a paper that was published in Nature just a month ago that suggested that at about the same time period in the Amazon in Bolivia, they were already also practicing agriculture. So there might be the possibility that another area of the world was the first place where humans transitioned into that. But this is where it shows you where all over the world domestication has occurred and where agriculture has occurred. So originally, as I said, the Fertile Crescent is believed to be where the oldest evidence of domestication is seen. And when there are many of our founder crops of the world came from. So wheat, barley, rye, oats, lentils, sheep and goats and cows were domesticated in this area, and chickpeas. But starting 12,000 years ago, it started here in the Fertile Crescent, but it occurred worldwide actually, in different places independently. So China also independently started to domesticate species in this area. And there is believed they appear to have done that independently. There's no evidence at that time point of cultural contact between the Fertile Crescent and China. And in China, what they started to domesticate, you'd be surprised. We'd think of Chinese civilization as depending on rice. They actually domesticated millets. Millets was the first thing they domesticated. In fact, early Chinese civilization was based on millets. And in the old world, the new world, they started to domesticate many other species as well. And in fact, that's why we know it occurred around the world independently, because 12,000 years ago, there was no cultural contact between the new and the old world. And in the old world, they also started to practice agriculture and domesticated several species, including corn, sunflower, potatoes, squash, and beans, and peppers. So domestication started to occur about 12,000 years ago during a period of time called the Holocene. Now, the reason I became interested in this is I'm interested in evolutionary biology and the process of evolution. And in order to try to understand evolution, evolutionary biologists study a wide range of different species, trying to understand how they've evolved over time. If you read Darwin's Origin of Species, his first chapter is actually about domestication. Darwin saw domestication as an important evolutionary process. He looked at domestication as a way to understand variation, which was a key component of the evolutionary process. He also used it as a way to show people that by the process of selection, in this case, selection under breeders, you could get a wide range of different breeds that may look very different from each other, even though they were one species. He looked at the process of selection in domesticated species as an analogy to natural selection, which he then proposed as a means of a major mechanism for the evolution of species in nature. And so he was fascinated by domestication. His first chapter in Origin of Species was about domestication. And the first book he wrote after Origin of Species was this book, The Variation of Animals and Plants Under Domestication, which he's published. The interesting thing is, I don't know if you know this, Origin of Species was meant to be an abstract. That the entire book is an abstract. He said he was writing this abstract, and then he would spend some time writing out the entire book. And Variation of Animals and Plants Under Domestication is, in fact, the first chapter of what Darwin called his big book. Unfortunately, after he wrote the first chapter, that is the first Variation of Animals and Plants Under Domestication, which is a 2-volume work, by the way, he gave up and he said, yeah, he couldn't, it was too exhausting to write the entire big book, so he stopped there. So since Darwin's time until now, domesticated species provide a nice model system for us as evolutionary biologists to study the evolution of species. And there are several reasons for that. One is that these are young species. All domesticated species on the planet, except for the dog, okay, has only evolved in the last 12,000 years. The dog is the exception. The dog was probably domesticated 30,000 years ago. But all other domesticated species, almost everything we eat, the cotton we use in our clothes, and things like that, only evolved in the last 12,000 years. And also because these are crop species, we know a lot about the genetics of these species. And so me as an evolutionary geneticist can take advantage of the knowledge that have been gained about the biology of these different species to try to understand how they've evolved. And there's a third thing that propels me to try to understand the evolution of these species, because these species, as you probably can appreciate, food security is becoming to be a major problem in the world and will continue to be a major problem going forward. And what I and several others who are studying the evolution of crop species hope is that by understanding how these species have evolved, we might be able to help breeders in the future in developing new varieties that are adapted to different environments. At least that's kind of the hope in the future. So in my lab, we studied 2 really major types of domesticated species. One here on left is rice. There are 2 species we study, Oryza sativa, which is Asian rice, and Oryza glaberrima, which is actually a rice that was independently domesticated in Africa. I'm not going to talk about glaberrima today. I'm going to focus on Oryza sativa. I actually have a lab in New York where we study rice. And I also have a lab in the NYU campus in Abu Dhabi. And there we study date palms, or Phoenix dactylifera, which is a different domesticated species that's important for the Arabian Peninsula. But today I'm going to focus my discussion on Oryza sativa. And the Oryza sativa, we're all from the Philippines. I think most of us are, all the ones watching are from the Philippines. We know how important rice is. It is a major food crop. It is the most important food crop in the world. It feeds more people than any other food crop in the world. It's a primary calorie source for approximately half of the world's population. We'll show evidence that it's domesticated in China. It's got a relatively small genome for as a eukaryotic species of 400 million base pairs. So that makes it easy for me as a genomicist to work with it. And as I said, we think, Oryza sativa evolved, and I'll show you evidence for that, about 9,000 years ago from a wild species, Oryza rufipogon. Oryza rufipogon, it's wild rice. It's not the wild rice that you buy in the supermarket, that's actually a different species altogether. It's not Oryza. The real wild rice, Oryza rufipogon, right now grows in Southeast Asia and South Asia. It used to grow all the way into northern China. And Oryza sativa was domesticated from its wild ancestor, Oryza rufipogon. I mentioned in Africa, they also domesticated a different Oryza species. Oryza glaberrima was domesticated in West Africa from Oryza barthii. So it's an independent evolutionary event. Where does rice grow? This map from the Food and Agriculture Organization shows you where rice is grown worldwide and the number of rice seeds is grown. As you can see, it's grown essentially in every continent right now except for Antarctica. But if you look at where most of rice is grown, this is where most of rice is grown in the world. In this area around the circle is where rice is the major staple crop for much of the population in that region. And it's also something to note that half of the world's population is within that circle. So half of the world's population is really dependent on rice as a staple grain. One of the things that many of us appreciate, probably, who are rice eaters, know is that there's actually a lot of variation in domesticated rice. Rice is not just a monolithic monotype, but there are several varieties and cultivars. In fact, at the International Rice Research Institute, they have about 120 or 130,000 varieties of rice in their seed germplasm bank that we as scientists use in our studies. So there's quite a bit of a range of variation in domesticated rice. And if you look more closely genetically at rice, there are about 4 major groups of rice, all within Oryza sativa. One is subspecies Japonica and the other subspecies indica. These are the 2 major variety groups, and they're actually subspecies. They're now recognized, well, they were recognized as subspecies starting in the 1920s. And we're going to talk about the history of these 2. There's also several other rice variety groups. They may be considered species, but they haven't been elevated to species yet. Circum Aus or Aus is a rice that's grown primarily in Bangladesh. It's genetically distinct from Japonica and indica. And Basmati, you're probably familiar with the fragrant aromatic rices of South Asia and partly Thailand. That genetically is distinct from Japonica and indica. But Japonica and indica are the ones that are the major groups that comprise most of the varieties that are grown around the world for Oryza sativa. So, you know, I started getting interested. Actually, I started getting interested in rice in high school. To be told, when I was a student at Manila Science High School. I went to Erie to do my National Science Fair project in rice. So I was interested in rice all the way back in high school. But professionally, I really started getting interested in rice I would say about 2000, 2001, when we started to, in my lab at that point, we were looking at the evolution of many different types of species. And I just said to the lab, you know, let's start to study rice because I'm interested in trying to understand the evolution of this domesticated species. And also, I'll tell you the truth, I wanted to use it as an excuse so I could go back to the Philippines as often as I could. That was one of the reasons why I also decided to study rice. Well, one of the things we earlier did, and this was also the time when genomics was becoming an important technology, and we were trying to understand genomic variation in many different species, the Human Genome Project and other species around the world. And so one of the things we did, which was, it's actually now, it's such a low scale project, but at that time it was almost state of the art. I said, well, we want to look at variation in the genome of rice. But at that point, at about mid to early 2000s, we still didn't have the technology to do whole genome sequences at a large scale. So what we did was we sequenced about 110 genes of rice in different varieties. So we had varieties that were aromatic Aus, indicas, japonicas. We also had wild rice, Oryza rufipogon, and some outgroup species, Rhizum marginalis and barthii. So we had about 110 different gene regions that we sequenced using old-fashioned Sanger sequencing. And basically we used that to do a lot of things. One of the things we did is we looked at the relationships of these different rice varieties. And when you look at these different rice varieties, you see that there are 2 groups of rice. Japonic and aromatic group actually groups together as one group, and they're genetically distinct from indica and Aush. which form another group, okay? And this suggests that there are two evolutionary lineages of domesticated rice, the indica lineage and the Japonica lineage. We were not the first ones to show this. This had been seen 20 years, maybe even 30 years prior to that by a variety of enzymatic studies, reproductive isolation studies, and some microsatellite and RFLP studies. So we were not the first ones to show this, but this was, I think, one of the first to show from data across a large number of genes across the genome that in fact these genetic lineages were distinct from each other. And because there were two lineages in rice, and this had been now known for about 30 plus years, what people had understood about the evolution of rice and where it came from was something like this. Oryza rufipogon is the ancestor of domesticated rice, and it was growing from China, India, and Southeast Asia. This is where Oryza rufipogon, or wild rice, grows. Oryza japonica evolved 9,000 years ago, probably in China in the Yangtze Valley. So that's where Japonica rice came from. Indica rice evolved separately. Oops. And by the way, I'm sorry, I'll come back to that. Japonica rice evolved 9,000 years ago, and it evolved starting in this period in climate history called the Holocene. And especially this is called the Holocene climate optimum. So if this is the temperature here on the left, colder, warmer, 12,000 years ago, before that was the Ice Age. So it was, it was very cold. And then it started to warm with the melting of the glaciers. And then starting about 9,000 years ago, the world hit what was called the Holocene climate optimum. And this is actually when much of the early domestication and the start of agriculture started around the world, was during the Holocene climate optimum. So it was around this time that in the Middle East, they were starting to grow wheat and barley. In China, they were starting to grow millets and some rice. Corn was being grown in the Americas. So it was during this time period. And for rice itself, there was a lot of different changes that occurred between wild rice, Oryza rufipogon, and sativa. So if you see wild rice, if you look at the density of the panicles, these are the branches that carry the seed. In Oryza rufipogon, there's a few of them, and they're very loose. They're much denser in sativa, so you can pack a lot more seed. The morphology of the seed is different. There's bigger grain in Oryza sativa. It's lighter. They don't have these awns, which are useful for seed dispersals. There's a lot of changes. There are only some of them that differentiate wild rice from Oryza rufipogon and Oryza sativa. And in the archaeological record, you also see this transition in archaeology of seeing a lot of wild rice to seeing domesticated rice. Okay, you can start to see domesticated rice grains start to appear in the archaeological record, starting about 9,000 years ago. And the process by which rice was domesticated actually took some time. About 20 or 30 years ago, if you ask evolutionary biologists how fast domestication occurred, some people thought domestication actually occurred in a very short span of time, 100 or 200 years, but it occurred very rapidly. That was the accepted paradigm in the study of domestication. We have shown subsequently by myself and others, and using archaeological data, that rice, for example, did not get domesticated in 100 or 200 years. It actually took about 4,000 years to domesticate rice. So this is a plot where it shows you the frequency of domesticated rice versus wild rice in the archaeological record in China. So about 8,000 years ago, there's about 20% of domesticated rice. And it takes until about 4,000 years ago before you get almost all of the rice being domesticated rice. So it took that period of time for it to reach what we in evolution call fixation, for wild rice, domesticated rice to become prevalent in farmers in China. And that's a period of time, I mean, that taking 2,000 to 4,000 years to domesticate a crop species is also seen in other crops. So this is data from the Middle East in barley and in wheat. Also, it takes between 2,000 to 3,000 years to domesticate barley and wheat earlier. So domestication as a process is not something that occurs fast. Some people say it occurs fast, and there's still some scientists, particularly some, shall we say, colleagues in Israel, who still believe it occurred very quickly. We think that they're the minority opinion now. I think people are beginning to come around the idea that myself and others have shown based on archaeological records and later on from genomic data that rice took a long time and other domesticated species took a long time to domesticate. Okay, so that's our Japonica rice, and it takes about 4,000 years for it to evolve. So indica rice, what happens to indica rice? Well, Japonica rice starts 9,000 years ago. Indica rice was believed to have evolved independently in the Ganges in South India somewhere between 4,000 to 8,500 years ago. Here, the archaeological record prior to 4,000 years ago is actually not very good for seeing a lot of indica rice. But definitely after 4,000 years ago, you start to see indica rice in India. And because as I showed you, genetically, indica rice and Japonica rice, while they're Oryza sativa, they are genetically distinct from each other. This had led many to propose that rice evolved twice, Japonica in China, indica in India. The first major indication that this might not be the story came about from a study, and because of this, by the way, this is how we think of rice happen, right? Japonica evolves in India, Japonica evolves in China, indica evolves in India, Japonica then spreads to East Asia, and then what happens is indica evolves in South Asia, and then it spreads both westwards into the Middle East and eastwards into Southeast Asia, and actually to China. After indica spreads into China, it replaces most of Japonica in China. Japonica is just pretty much grown in Asia, in Japan and Korea, and parts of Southeast Asia. It's mixed between indica and Japonica. So that's a story of the evolution of rice as we thought about it maybe 10 years ago. So let's fast forward a little bit to a study we did about 2010. And at this point in time, remember the previous study I showed you where we sequenced 110 genes. Here, we were becoming a little more confident. And what we proposed here, instead of just 110 genes randomly scattered across the genome, we proposed and got a grant to sequence genes on 3 chromosomes, chromosomes 8, 10, and 12 of rice. And we chose these because these were some of the smaller chromosomes in rice. And we proposed to sequence a gene every 100 kb across the rice genome, across these 3 chromosomes. So instead of 110 genes, we were now looking at about 500 to 600 genes that we sequence along these 3 chromosomes to see how variation changed along a chromosome. So we started this study, and this is kind of the data. This was published in PNAS. Here's chromosome 8, chromosome 10, chromosome 12. This is pi, which is the level of nucleotide diversity. You see how it varies across a chromosome. There's some variation here. There's no variation here. Lots of variation here. This is the centromere, which is involved in the cell division, in chromosomal assortment. There's very little variation and so on. Okay, so that's the level of variation we see. And then we saw this as an opportunity to use all of this data to study the evolution of Japonica and indica. And the way we did this is the following. So one of my collaborators in this project was Carlos Bustamante. Carlos Bustamante is then he was a professor at Cornell. He's now a professor at Stanford. And he's one of the premier theoretical population geneticists. And what he specializes in is taking genomic data and using the amount of variation in genomic data to infer something about the evolutionary dynamics that have occurred in that species. And one of the programs or one of the methods that he developed was this program called DADDY. DADDY stands for Diffusion Approximation for Demographic Inference. And it's based on this equation you see here, the scary equation here. This is actually the Kimura diffusion approximation equation for neutral sites. Okay, so what this is that when you get a mutation that occurs in a population, it will increase and decrease randomly in the population in a process called genetic drift. The dynamics of its increase or decrease in frequency can be approximated by a diffusion process. And this is Kimura's famous diffusion equation, where he says the change in frequency of a mutation in a population over time is dependent on the frequency of that mutation when it starts, It's dependent on selection. This is selection. It's dependent on migration, that is the ability of genes to come in and out from other populations, or that mutation to come in and out of other populations. And it's dependent on a mutation rate. Okay, so this is the diffusion approximation. And what Carlos basically did is he developed a method by which he could estimate the parameters in this diffusion equation if you specify a model and given some data about the frequency of different mutations across the genome of a species, in this case, the rice genome. So since he was a collaborator, said, let's test this. And we said, let's test two models. The model here on the right is the model that was the accepted model for the evolution of rice. And the model goes like this. There's wild rice, the Oryza rufipogon, and sometime in the past, a tau, you have the evolution of Japonica. So this group here is Japonica, it's evolved. And then later on, independently, you have indica come out of Oryza rufipogon. And there's some gene flow between these. There's some period of time where that evolution is taking place. And so that's the model that we propose. And what Carlos can do is apply the diffusion equation and using the variation from the genomic sequences we got, or the gene regions we got, to estimate all these parameters in the model. When did it occur? And how much migration was occurring, and so on. And the other thing is then test, is this model likely, or other models more likely for the evolution of rice? And I'm still not sure why we decided to do this, but we decided to test this 2 origin model to a 1 origin model. And this model says, here's the origin of rice. And then sometime in the past, rice evolved. And then, but it was just one rice, and then Japonica and indica separated later on. Oops. Okay. So those are the 2 models we tested. So, this was a collaboration between myself, Barbara Shaw, who is a famous evolutionary geneticist at Washington University, Scott Jackson, who is a professor at the University of Georgia, and Carlos Bustamante. Carlos was the theoretician. And we had this big lab meeting at Washington University. I was the leader of this big collaborative NSF grant to study this. And Carlos presented the work that they had done analyzing this data. Don't ignore the top. That just shows you the observed versus expected frequencies of mutations. But here on the bottom is a table where we're testing the single founder or the double founder model, okay? And on the right is the likelihoods of these 2 models. And I like you to concentrate on the ones with the arrows here. See that the likelihoods of the single founder model is actually higher, significantly higher than the double founder model. So when he presented that, I said, this can't be right. This is wrong, because everybody knows rice evolved twice. But you're telling me that the genomic data is showing that the model showing that rice evolved only once is outperforming the 2 origin model. And I said, Carlos, you can't, you've got to be, you're wrong. I said, I'll tell you why you're wrong. You're wrong because you didn't include selection in your model. You were dealing with it as neutral. So why don't you include selection? And he includes selection, and it still shows you the same thing. The single origin model is outperforming the 2 origin model. And at that time I said, Carlos, this is just, I just can't believe this is true. And so we broke up, me kind of shaking my head and thinking something's wrong here. Now, the one who actually, the postdoc in the lab was heading this project was Jean Molina. And this just basically goes to show you why I didn't believe it. We're showing genetically that indica and Japonica are genetically distinct. And it makes sense to think that these evolved twice. How can this be right? So Jean Molina was the one who was headed this. Jean Molina is from the Philippines. She got her PhD at Rutgers University in plant systematics. And she's now a professor at Long Island University. And she came to me and she said, Michael, what I want to do is, you did this phylogeny of Japonica indica showing that there's support for 2 origins here. And there's a lot of other studies, molecular phylogenetic studies that show 2 origins. Why don't I look at that data, this data and other data, to see if this is really true? And I remember telling Jean, don't do it. I mean, don't bother Carlos is wrong. Carlos Bustamante is a MacArthur Genius Award winner, so everybody really kind of worships him. But I said, Carlos is wrong here. He can't be right. But Jean didn't listen to me, which was good. And why she didn't listen to me is the following. About sometime that year, or that year that we were looking at this data, there was a paper published that said there are sometimes, there are circumstances when you estimate the phylogeny of a group of rapidly evolving and closely related species, that you will misestimate the phylogeny. And the way this happens is because when you're dealing with rapidly evolving species, what's happening is that you use data from multiple genes, okay? When you're trying to look at the phylogeny of a rapidly evolving species, you don't use data from only one gene. You use 10, 20, 100 genes to estimate your phylogeny. But because every gene by itself doesn't have enough mutations or signal to estimate the phylogeny, after all, these are rapidly evolving species or very closely related species, what you do is what everybody does. You take all of this, let's say 100 genes, and you put them all together and pretend that they're one gene, and then estimate your phylogeny that way. And this paper that just came out that year said, you know what, if you do that, and I just put the quote here, the concatenation approach, that is putting genes together under conditions in which a coalescent produces a high level of discord among individual gene trees, basically when you've got rapidly evolving species, leads to incorrect statistical estimation of phylogenies. What this was saying is that what we were doing under the circumstances, there was a good chance you were getting the wrong answer. Not only we, but everybody else who did what we did in rice phylogeny. So what Jean did was she took the same data that had been published by us and others, and she applied a different method. It's called BEAST, Bayesian estimation for sampling trees. And what BEAST does is it takes multiple genes, it takes the data from multiple genes, but it doesn't squish all of that data together and pretend it's one gene. What it does is it estimates the tree from each of those genes, even though the signal is weak, and then integrates the signal after it estimates the phylogeny. So that's the difference. It doesn't put the data together and then estimate one phylogeny. It estimates all of the independent phylogenies and then integrates it together in a Bayesian framework. And what Jean showed was really beautiful. She said, look, these are all on the left here in this table. These are all previous studies that had all shown that Japonica and indica evolved independently, separately. And this was the number of genes in each of these studies, 4 genes, 3 genes, 11, 20, 22, 5 genes. And when she applied BEAST to this same data, in 4 out of the 6 cases, actually the single origin model was strongly supported. The 2 cases in which single origin was not supported, actually it couldn't tell which model was good. So what Jean had shown was that we had all been looking at this very differently and in the wrong way. By both Carlos Bustamante's analysis of the population genetics and this new analysis, it became clear that what probably happened is that rice evolved or was domesticated once, and that indica probably came from Japonica. And the reason we thought that is if you look at archaeologically, Japonica is actually older archaeologically than indica. And when we wrote this up in 2007, everybody hit the roof. We got in BBC, South China Morning Post. I love this. U.S. study backs China and the Great Rice Debate. And in Jakarta, U.S. study on origin of rice sparks Sino-India row. So it created a lot of contention within the community. Because what we had suggested in that paper is this is what we think have happened. Japonica evolves 9,000 years ago in the Yangtze Valley, China. And we know that because of archaeology. In India, about 8,000 years ago, and a little bit after that, they were also dealing with Oryza rufipogon, wild rice. They were harvesting it, or they were beginning to domesticate it. They may have been starting to plant it. It had not yet been domesticated. That is, it was still kind of wild, but it was starting to get there. So it might have been a proto-indica. And then Japonica spreads. And then what we think happens is that Japonica then moves to India. it hybridizes with what was going on in India. So Japonica hybridizes with what was going on in India, and that gives rise to Japonica, which spreads. Okay, so we think indica is a secondary species that derives from getting genes from Japonica going into India sometime later on. And there's reason to believe this. If you look at archaeologically at rice in China, what you see is that rice actually, you start to see rice, domesticated rice, starting about 7,000 BC, as I said, in the Yangtze Valley, here in the lower and also sometimes somewhere in the middle Yangtze Valley. You start to, in the upper Yangtze Valley and the lower Yangtze Valley, you start to see domesticated rice. And it has a long history starting about 9,000 years ago. In contrast, if you look at Indian archaeological sites, you actually don't see rice in any quantity until about 4,000 years ago. It was only after 4,000 years ago that you started to see a rapid explosion of rice in India and its expansion throughout South Asia. And in fact, what happens is that the explosion of rice in India, starting about 4,000 years ago, is associated archaeologically in the northwest corner of India with pottery and tools that look like they were Chinese influenced. So that gave us that idea that maybe what happened is that Japonica had evolved in China, and about 4,000 years ago, Japonica made its way into India, into the northwest corner. It hybridized with what was going on there, and that gave rise to indica. And in fact, this was tested, quantitatively by a colleague of mine, Dorian Fuller, at University College London. He's a colleague of mine who we collaborate. He's an archaeologist and I'm a geneticist. And he used archaeological sites and the dating of archaeological sites to test two hypotheses: whether rice came from China via the southern route, or whether it took the Silk Road 4,000 years ago, or the Proto-Silk Road. The Silk Road is very old, or at least there was a route. It's called the Hexi Corridor, and went into India, the northwest corner. Some of us may think that this is the easier way to go down Southeast Asia and cross over into India. It turns out there's a lot of mountain ranges here that make it very difficult to cross from China into Southeast Asia, and then from Southeast Asia into India. So there's actually a barrier here. So he actually tested this with archaeological data, and this was published 2 years ago in Holocene, where it looks like that the southern route was less preferred than the northern route. The northern route, or the Hexi Corridor, in their parameter estimation, was the more likely route by which rice took from going from China into northwest India. Also, although a lot of people, when we published this, were still arguing, and we were feeling a little bit alone in our thinking about this, then the Beijing Genome Institute came and published this article in rice. They sequenced about 400 different rice varieties. And what they did was quite clever. What they did was they looked at all the genes that were important in the evolution of domesticated rice versus the rest of the rice genome. And they actually showed, based on their work, that the model we proposed was similar to a model that they also thought of, that rice originated in China. This gave rise to Japonica. It hybridized with something in India that gave rise to indica and aush, while ancient Japonica gave rise to temperate Japonica. The only difference is that they believe that rice was domesticated in the Pearl River Delta near Guangzhou, near Hong Kong. That archaeologically, that doesn't make sense. The Yangtze Valley is really where the oldest rice remains are found in China. So this southern China origin is probably wrong, but we believe that they found their domestication. And recently, we actually did a study where we showed from whole genome data, using whole genome sequences, that in fact there's evidence of 15 to 17% of the indica genome really coming from Japonica, being introgressed from Japonica. Again, providing genome support for our idea that what we think is that what was happening is that these genes that were important for the evolution of domesticated rice, things like that involve the ability of rice not to scatter its seeds or to grow upright instead of sprawling, that came from Japonica rice. And so the model we have is Japonica evolves in China, and then it moves into India. And then what it does is it brings all of these important genes that were important for the evolution of domesticated rice into something that was happening in India that gives rise to indica. Okay, so that's kind of the model by which we're proposing. So in the last 20 minutes, I'll show you something that's very new. This is a study that we've just completed. It's actually coming out in Nature Plants either the next 2 or 3 weeks, sometime very soon. We still don't know when. But really it asks the question, okay, now that we start to understand how rice originated, how did it actually spread? Okay, and the spread of crop species is an interesting problem, because think about where crop species evolve. Wheat starts here, rice starts here, but after a few millennia, they've dispersed through a wide areas around the world. And in doing so, they have had to adapt to a lot of different environments and habitats. So for example, rice coming from Yangtze has to evolve to be in higher latitudes in China and in Japan and Korea. It has to go to near equatorial regions in Southeast Asia. It has to deal with different cultures. It has to adapt to different environments. And it has to also adapt to different biotic interactions, such as different pathogens and insects. And so, trying to understand how crop species are dispersing after they've evolved might give us clues to how these different crop species are able to adapt to different environments as they're dispersing across the world. Okay, so that's the kind of idea. And this is actually the main area of research that my lab is interested now in the next few years, is to try to understand this process by which these different crop species have moved out of their origins and have adapted to different environments. And archaeologically for rice, we kind of have a broad archaeological record. So you might not be able to see this. You're going from top left to right, going this way. This is where rice archaeological remains starting about 9,000 years ago. It's in the Yangtze Valley. As time progresses, it expands in East China, stays there until about 5,000 years. And then it starts to get out of this area between the Yangtze Valley and the Yellow River down south into South Asia and Southeast Asia, and move into Japan and China. And then, of course, in the present, Japonica rice is now found like everywhere in Asia. So archaeobotanically, we kind of have a record for this. And the question is, does the genome tell us something about the pattern of movement of rice and also the timing at which this has occurred? And to do this project, we used 2 data sets. There was an international consortium that China and the International Rice Research Institute did to sequence 3,000 rice varieties from the Erie Germplasm Bank. 1,265 of those are what we call land races. Land races are primitive, we call primitive, they're traditional varieties. These are varieties that have not been subject to scientific breeding. Okay, you might call them heirloom varieties. But these are things that were not bred by breeding stations or by public or private breeders. So these are traditional varieties. And so when we try to study the evolution of crop species, we focus our attention on these traditional varieties of land races. In the 3,000 Genome Project, there were 1,265 land races that had good geographic information. That is, we knew where they came from. In my lab, we also sequenced about 178 different land races. So we had that data to add. So this was what we used. This was whole genome sequencing data using Illumina short read. And I think each land race was sequenced about 12 to 14 times. And we developed, I'm not going to go through this. This is a whole pipeline of how we got the data. We basically, from this entire set, we called about 41 million mutations in the genome. And then we use a variety of filtering steps to come down to sets of SNPs or single nucleotide polymorphisms we could use in our analysis. Okay, and the first thing we did is we took all of this variation in land races. And by the way, here's a map of where these land races are found all over Asia, both indica and Japonica land races where we find. Notice that we don't have a lot in China. Some of this, I'll tell you later on something. China actually, it's not really well sampled, and we'll come back to that later on. But we have about 1,400 land races, and we did a principal component analysis to show you genetically what those millions of mutations, what it tells us about the relationships between these different land races. And what you see here is, in general, you see 4 clusters. This cluster here is the indica cluster. This is indica. Here on the upper right, this is Japonica. Down here is Aus. Here it's a little bit diffused. This is Basmati. And then there's a whole bunch of things. Okay, so there we go. This is Japonica land races. This is the Japonica land races here. This is 372 Japonica land races. If we take this group and we blow it up, we can start to color it from based on the country in which they're found. And you can start to see that this is something that actually surprised me, that the rice traditional land races still had a geographic signal from where they came from. So rice from Laos pretty much grouped together, rice from Japan grouped together, Indonesia, Philippines grouped together, and so on. I was surprised by that because I thought, well, maybe people were moving rice around their region or trading it or moving it so that we won't have a geographic signature in the genome, but we're seeing a geographic signature in the genome. So we're able to see different groups of traditional varieties and where they're found in this space of genetic variation. And what we can see is they group into different populations. Here you have a group of Northeast Asian rices in Korea and Japan. These are the temperate rices. There's another group of temperate rices we'll get on later here. This is mainland Southeast Asia. We had good sampling from Laos and a little bit from Bhutan, which we think of, for the purposes of this talk, let's call that mainland Southeast Asia. Then we have the island Southeast Asia. So we've got groups in Indonesia and groups in the Philippines are grouped here. And you can see here the map, the color coding of the different groups of Japonica rice that we have and traditional varieties. So these are the populations. Now, what we did is we can then see how these populations are related to each other. And the way to do that, there's several ways to do that, but the one that's used heavily in human genetics and human genomics is what's called admixture graph reconstruction. And that is try to reconstruct the relationships between these different populations, including possible migration or hybridization between populations in this group. And so this was started by a postdoc in the lab, Rafael Gutaker, who at the end of summer is leaving to be research leader at Kew Gardens. He was the one who really led this project. And what he did is he did the graph reconstruction. And this is the graph reconstruction. It shows you the relationships between these populations. So you have an ancestor here, an ancient ancestral population here. It splits between the group of rice that grows in Northeast Asia versus essentially the rest of rice that grows in Southeast Asia. And you can see the various relationships here shown in solid arrows. Where you see dotted arrows are evidence or estimates that these populations are a hybrid between different populations. So one example here is this Philippine population here. The ancestor of this Philippine population appears to be a hybrid from another population in the Philippines, another population in the Philippines, as well as some ancestral ancient Japonica population that we have not identified yet, okay? So that's what those dotted arrows are. So this kind of gives you the reconstruction of what the relationships of these populations are. But the other thing we can do is we can time the timing of when these populations separated from each other. And the way you do that is a method called the cross-coalescence. It's basically taking the mutations between two different populations and looking back in time as to when genomic regions start to what we call coalesce, or essentially essentially their sequences coalesce because you can trace their mutations back to an ancestor. And you can use population genetics to time when these populations coalesce. And we were able to time different places in this reconstruction that was important. And I'm going to go through this reconstruction one by one so that you'll see what we mean. So this is the story of what we think happened in Japonica rice to the best of our ideas right now. And this reconstruction is based largely on genomics, but it's also based on consultation with archaeologists who basically looked at our data and gave us and gave support for this model of how we think rice evolved. So 9,000 years it's domesticated, actually fairly far north in China, in the Yangtze Valley. And it grows, we think, for 4,000 years in this region between the Yangtze Valley and the Yellow River. There's a split in the population. So the one group of rices goes to Southeast Asia that eventually reach Laos and Bhutan. The other goes to northern China, and especially Korea and Japan. This is the temperate rices in Korea and Japan. This is the tropical Japonica that goes down south. And also there's in Laos, there's this upland tropical Japonica that again is coming from that split. We are able to, and if you look at the phylogeny of rice from our earlier study, you actually see there's a genetic separation between temperate and tropical Japonicas. You can see that difference between these two groups of rice. And we can time that split. That split appears to occur during a span of time between 4,100 and 2,500 years ago. That's when we think the separation of tropical and temperate Japonica actually occurred. Okay, so what was happening during that time? This is based on a large different number of population genetic work. This is the time when we think the timing of the split occurred, sometime between 4,100 to 2,500 years ago was when temperate and tropical Japonica separated. Here at the bottom is something interesting. We collaborated with another archaeologist who also modeled climates in the ancient past. Her name is Jade Guedes, and her collaborator Kyle Businski. Jade Guedes is in the University of California at San Diego. And what she had shown prior to that was that what happened in China was that in the past, actually, the climate was actually warmer. The temperature was warmer. But sometime about 4,200 years ago, what happened was there was this event that's been called the 4.2K event. This was a climatic event that resulted in a dramatic cooling in the world, where temperatures all around the world actually started to drop and actually have maintained that lower temperature. It was about a 0.7 to 1 degree drop in temperature starting about 4,200 years ago. This 4.2K event actually had a number of dramatic consequences around the world. It actually, there was a period, it was a period of agriculture turnover in the Middle East. And also in China, this was a period of rapid destabilization because of a large number of famines that occurred in China starting about 4,200 years ago. And Jade did something that was actually really interesting. What she did was she took tropical Japonica and she asked the questions, tropical Japonica needs a certain amount of temperature in which to grow. It needs a certain amount of temperature over a certain span of days. It's called the growing degree days. And what she did was she reconstructed the temperature all over Asia in the past and said, based on that temperature, where can tropical Japonica grow? And as the temperature changes over time, where's tropical Japonica able to grow? And so this is the map about 5,500 years ago, okay? This is where tropical Japonica can grow. And pretty much it can grow everywhere in Asia. It can grow all the way up into, into Russia, into Japan and Korea. It's able to grow there. It's only the Tibetan plateau that it's unable to grow. So this is a movie. I'm going to show you, I'm going to run this movie. And when you start to see dark blues, that's when tropical Japonica cannot grow in those regions, okay? So 5,500 years ago, this is what it is. So we run the movie. And 5,000, 4,900, 4,800, 4,700, 4,400, 4,000, oops. Let's try it again. It's growing up. And then the 4.2K event begins. And you can start to see, look, tropical Japonica can now only grow in Southeast Asia during this period. Okay, after 4,200 years ago, tropical Japonica can no longer grow where it originally was seen to be growing. And today, in the present, tropical Japonica can only grow in Southeast Asia, both island and mainland Southeast Asia. So that's what happened to tropical Japonica. And archaeologically, you see that happen. Rice is growing in this northern area in China between the Yangtze and Yellow River for between 9,000 and 4,200 years ago. After about 3,500 years ago, where you see archaeological rice remains are in Southeast Asia and further north in Korea and Japan. Actually, a lot here, they're starting to grow wheat from Europe. But that's where you're starting to see from the archaeological records. So it makes sense. So what happens 4,200 years ago? What happens is that because of this cooling event, rice is forced to go south into Southeast Asia. It's forced to grow south into Southeast Asia. And this actually coincides with migration of farmers from like the Yunnan area and that area in China southwards into Southeast Asia. This is based on ancient DNA studies of humans. Also, we know from archaeological studies that rain-fed rice agriculture occurred at about the same time in mainland Southeast Asia, and that we're beginning to see metallurgy, metal implements from the Yunnan province in China go into Southeast Asia starting about 3,500 years ago. So we see from the paleoclimate reconstruction, we see from our genomic data, we see from ancient DNA data of humans, and we see from archaeological data of rice remains and artifacts in Southeast Asia that rice appears to have moved into Southeast Asia starting about 4,000 years ago. So the split of tropical and temperate Japonica takes place about 4,000 years ago. So how about the spread of rice into Southeast Asia? Starting about 4,200 years ago, we see a split into island Southeast Asia, but the timing of that split actually occurs a little bit later. When we think rice starts to go into Indonesia, Borneo, and the Philippines, is actually in a time period between 2,500 and 1,600 years ago. That is this, that shows you, actually that's wrong. That's actually this split here, which shows you the movement into these different islands and into island Southeast Asia. We're beginning to see these estimates crop up. This is an interesting time because what you see is that during this time period, there's an intensification of rice production all over Southeast Asia based on the archaeological record. And then what also you begin to see in island Southeast Asia, that is in Indonesia, Borneo, and the Philippines, you're beginning to see these special drums that were being made in Vietnam start to disperse really rapidly throughout island Southeast Asia. And archaeologists now think of this as what is called the Austronesian trading sphere. Starting about 2,500, 3,000 years ago, there was a lot of movement of trade in this area. And we think starting about 2,500 years ago, rice began to go into the island Southeast Asia from mainland Southeast Asia into based on our genomic data. The rice from the Cordilleras, the Ifuga rice, it's very interesting. It turns out it appears to be a hybrid between that the introduction of rice, we think from Borneo, from mainland Southeast Asia, starting 2,500 years ago, and another unknown lineage that's more ancient from mainland Southeast Asia. So it's between this old population and this this introduced Philippine population that gives rise to the Ifugao rice. We don't know when this occurred dating wise, and we don't know where this ancient lineage comes from. We actually have gotten sequence from Aboriginal rice that's grown by Taiwanese Aboriginal peoples in Taiwan. Maybe that might tell us something about this, but it's not yet clear. Because I'm really intrigued by the possibility that this ancient lineage is a lineage that comes from Taiwan when people began to move out of Taiwan into Southeast Asia and Polynesia, starting between 3,500 to 5,000 years ago. But we'll see from the genomic data whether we can see that. So stay tuned. Then there's another group of rice in Japan, the upland temperate Japonica rices. This is not irrigated rice. This is grown up in the hills and mountains. This is rain-fed, and this also appears to be a hybrid between some rice from Southeast Asia and a temperate group in the north. We might study that as well. So that tells you this is what we think the model of the movement of rice and the dating of when this occurred or has taken place. This gives us now a framework to try to understand how rice has dispersed all over Southeast Asia and all over Asia. Now, indica rice is a separate question. We also tried to reconstruct indica rice. We're able to look at different populations of indica rice and reconstruct its admixture graph. But the problem with indica rice is that the admixture graph reconstruction was pretty unstable. From modeling to modeling, it would not give us branches that were stable as we increase certain parameters in the modeling. And we think this is because of a number of different things I won't get into right now. But in fact, the only thing that we were always able to be sure of is in indica rice, when rice left India to go to China and Southeast Asia, this split here, that was always stable in our reconstruction. So we were able to date that movement of rice. And it actually occurred fairly late, between 1,400 and 2,000 years ago. That's when we think rice moved out of India into China and Southeast Asia. That's the only thing that we have with some certainty. And that's interesting because starting 2,000 years ago, guess what? That's when Buddhism moves to China from India. So there might have been more movement there and the movement of indica rice. And by the way, indica rice is the rice that's now primarily grown in China, and it's actually a pretty dominant rice in Southeast Asia, including the Philippines as well. Japonica rice is really only largely grown now in temperate regions of Japan and Korea. Although in the Philippines and Indonesia, there's also some Japonica rice that's grown. Oh, again, and indica rice comes into central Thailand archaeologically fairly late as well. Okay, so I'm sorry I went a little bit over, but this gives us models now for the dispersal of rice in Asia based on Japonica and indica rice and how that's moved. The whole paper is coming out in the next few weeks in Nature Plants, so stay tuned for that. You'll be able to see it in all its detail. One of the issues we've had as we're going forward, so this is kind of in the future, is if you look at where this is Japonica rice and where we have our samples, you'll notice that China doesn't have much sampling. It turns out that when I ask my Chinese colleagues, they say they don't have Japonica land races in China now. Everybody uses indica, so that there are no Japonica land races in China. And also, as parts of Southeast Asia, we actually don't have very good sampling. We have very good sampling in Laos, in the Philippines, and Indonesia. But for example, Thailand, Cambodia, parts of Vietnam, and Myanmar, we have relatively poor sampling from that. So, the problem is a lot of the traditional varieties where we have access to the seed, they don't come with good information on where precisely they came from. In order to do this study well, you need to know precisely where these varieties came from. So Rafael Guttaker, my postdoc, had this brilliant idea. So instead of going to the seed bank like Erie to get seed, why don't we go to the herbarium, where collectors and explorers have collected rice in the 19th and early 20th century before the Green Revolution, and get DNA from the leaves in this herbarium. So these are specimens that are 150 years old, and you can get DNA out of them and use it to sequence their entire genome of these old traditional varieties. And the beauty of these things is that they actually tell you precisely where they were collected. So you're able to get good collection information. So in the past 6 months, Rafael has gone and collected tissue from herbarium specimens in New York Botanical Garden, Harvard Herbarium, Smithsonian, the Natural History Museum in London, Kew Gardens. We still have to collect in Paris and maybe in Field Museum in Chicago. And in fact, right now, what we have samples, we now have, as you can see, we now have more samples. The ones in the yellowish green are the new samples we have from herbarium specimens. Now we have better sampling in China. We have a little better sampling in Vietnam, Philippines. We have some in Myanmar and so on. So we're going to be able to advance this work using this herbarium sampling to get an even better picture of the evolution of rice in Asia. And so why are we doing this? We now tell the story of how rice evolved. Why are we doing this? Well, one is we're interested in the evolutionary process and telling the story of how this important species has spread and evolved around the world. But the other thing is as this species has evolved across different parts of the world, it has had to adapt to different climates and different environments. And one of the things my lab is interested in doing is trying to understand what genes were responsible for this traditional varieties to adapt to these different places around the world, using our understanding of their evolution in the past, in past climate change, and maybe to help breeders develop new varieties that may be important in adapting to other environments or even to future climate change. That's the grand hope. We have a grant now from the Ziegler Family Foundation to in fact explore this idea that by understanding the past, we can try to see how we can help the future. So just to summarize, we think domestication of rice was a protracted process, was not sudden. It took about 4,000 years. Hybridization and integration are important in the evolution of the species. The importance of the evolution of indica as a hybridization for Japonica rice, and that the major impetus for the dispersal of Japonica rice took place 4,200 years ago with a climate cooling event called the 4.2K climate event. Just to wrap up, we have other projects in the lab that you might be interested in looking up. We have a study on date palm and the evolution of date palm and the genome diversity in the Middle East and in North Africa. We have other more functional projects. We have projects where we're trying to look at genomic information to try to understand how mutations in the rice genome might be important in fitness or yield. And there's a paper that we published in Nature Plants in February that talks about the rice fitness consequence map project. We had a project looking at selection on gene expression in rice that was published in Nature in February also this year, where we're able to look at how strong selection is acting on gene expression. We've been involved in trying to look at how genes are connected in networks to each other, especially in response to different environmental stresses such as drought, such as salinity. So this is something that we're working on. A graduate student in my lab has been looking at structural variation in rice genomes, and she's been working on that in the last few years. And all of this, the projects in the lab, as I said, is really paying homage to Darwin. Darwin was one of the first to really champion the species as a way to understand evolution. We think we also study evolution, looking at these domesticated species, and also more importantly, or as importantly, not only understand evolution, but use the lessons of evolution to try to see if we can help breed new varieties for the future. So with that, I'd like to thank all of my colleagues and collaborators. The major people I want to thank is Jay Choi and Rafael Gutaker, who led some of the major issues that I talked about, the studies I talked about today. Jean Molina and Ana Caicedo have already left and have their own faculty positions, one in Massachusetts, one in Long Island University. There were more in past studies. These were all the collaborators from the studies past and present, and we're funded by these rice studies are funded by the Ziegler Family Foundation and the National Science Foundation Plant Genome Program. Oh, and these are past members of the Purugganan Lab and other collaborators. So with that, I'd like to leave you with a cartoon about the domestication process. And I love this one. This is a wolf looking around and saying to himself, I'm cold, I'm hungry. I see humans around a bonfire. Maybe I can ask for food. What could possibly go wrong? 30,000 years ago, this wolf thought that. And today, 30,000 years later, this is what happens after you domesticate something. So thank you very much for your attention. I'm sorry I just talked nonstop and fast. I hope that was understandable, and I'm happy to answer any questions people may have.
Wow, thank you so much, Michael, for that talk. It really felt like I was watching a documentary from Discovery Channel. So we have
Thank
a few,
you.
yeah, we have a
Right.
few questions in the chat box. So for the
Okay.
others who are still not familiar with the chat function, so you can find it at the bottom of Zoom. So if you have any additional questions, so I'll just take some of the questions there, and I think Joel might have one.
I do.
Okay, so let's take the first one first from Gary Cheng. He actually has two questions. One is commercial, while the other one is more philosophical.
Right.
Michael, what do you think about Henry Lim's SI Agritech's rice hybridization program? They have
I'm sorry, what a rice hybridization program?
Henry Lim's. I'm sorry, but
Oh,
yeah, but Gary already left the Zoom meeting because he had to hop on to
yeah.
another meeting. But he said he'll
I'm
check on
not
the YouTube.
familiar. I'm not familiar with that.
Okay, but he
With
said
that study.
additional information. He said that they have successfully produced hybrids that improved rice yields from a national average of 3 tons per hectare to over 7. It can potentially be a game changer. Maybe you can comment on common
Yeah, I
programs.
wonder.
Yeah.
Well, there's a lot. Obviously, there's a lot of work on increasing yields in rice.
Mm-hmm.
I'm wondering if this is the hybrid rice project that, of course, China is advanced in. But I'm not particularly familiar with that specific
Mm-hmm.
project, primarily because I'm not a breeder. I'm actually an evolutionary biologist. But certainly increasing yields are important for the future, because in order to try to feed the world in 50 years, we're going to have to need to double current yields of all of the major food crops. So increasing yield is going to be very important. But the other thing I'd just like to point out is that, especially in China right now, in that discussion with colleagues there, they're really now more interested in quality rather than quantity of rice. They're more interested in quality traits right now.
So what are they looking at, like in terms of quality?
A lot of different things. Aroma, they're interested
Okay.
in seed quality, in grain quality. But that's my colleague in Wuhan actually, which is a major rice program. That's what they're focused on.
Okay, he has a second question. Can you comment about Yuval Harari's thesis that domestication during the Holocene actually made life harder for Homo sapiens by tying them to land, creating more work
So
to feed an increasing
no,
population?
this is actually kind of an. So this is the one thing that people don't appreciate. Actually, we were better off as hunter-gatherers than we were as agriculturalists. If you look at the archaeological record, after agriculture started, people started to get a little bit shorter. You can see evidence of malnutrition, iron deficiencies, so many different things. So it's not clear. So in many respects, we were worse off. What appears to have driven it in part was because you could get stable food supply. The quality of the food might not have been as good, but it was stable. So and if we think about all of the things that happened after you get agriculture, you actually had to get you stopped being a nomad. You had to stay in the land. You had to stay there. There was increased density, epidemics, which we're going through now. We know how important density is. That really only became prevalent when cities started to evolve after the agricultural revolution. But then we got things like writing. We got, organized hierarchies might not be such a good thing. But we got writing come out of that and other things as well. So I think it's, we are where we are, and we just have to make the best of it.
Okay. Yeah. So maybe we can move on to Joel's question before I move on to the other questions.
It's a quick question anyway. Michael, thank you. It's a very fascinating presentation. So I was really eager to understand the evolutionary pressures that cause the divergence of indica from Japonica. And you explained it was a matter of hybridization, which is fine. But then later on, you talked about the 4.2K event, which was a climatic change, a cooling of the climate around 4,200 years ago. And if I am correct in recollecting it, that was about the same time period when indica diverged from Japonica. So could it have been possible that the hybridization that took place that led to the formation of indica from Japonica being precipitated by that climate change 4,200 years ago?
That we don't know, but of course that's an interesting coincidence in time. It's also, if you also look at India, rice was a northern crop in India until after 4,000 years ago. Then it goes south rapidly, very rapidly, actually.
Right.
So it may be, but we've asked Jade, my collaborator, is right now doing a better. Hopefully, she's going to continue to reconstruct climates, and maybe we can look at what's happening in India. There's also there was another thing. What I didn't talk about in terms of the paper was some of the movement in indica, and I believe into island Southeast Asia. One of the evolutionary pressures was actually precipitation. It was actually rather than temperature, it was precipitation. So the once it got there, then the diversification there was really driven by precipitation. In our paper, we talk about that. And in fact, that may be actually important. Well, it's an evolutionary pressure after 4,000 years ago.
Right. But I thought that was a very interesting coincidence.
Yeah, it's a really one of the problems in this field is you can see these coincidences, and I can make up a story, which frankly, a lot of the stories it could be completely wrong, right? Because the dates are right, but some of the things like Is it the Vietnam Dong San's group going into the island Southeast Asia? Was that what brought rice in? We really don't know, but
Right.
it's a coincidence in timing as well.
Thank you for that.
Just one quick thing, Michael. I got to meet Rod Wing when he was here in Arizona.
Oh
but
yeah,
I know that he's now at KAUST.
I know, and he's a good friend and collaborator actually.
Yes.
Okay.
Thank you.
Our next question comes from Mohanapriya from Tamil Nadu Agricultural University. He says that we know that rice is a self-pollinated crop. Is rice initially a self-pollinated crop during domestication, or did it get its self-pollination ability somewhere in the middle during the evolution?
You know, that's a great question. We don't know the answer. So the wild species is outcrossing. Rice is self-pollinating. Actually, a lot of domesticated species are self-pollinating for the most part. Not all, but for the most part. And we don't know when it occurred. So that's actually an interesting area of research. We know what are the important traits that are important for domestication, but it's not clear at what time point they appeared. What was the sequence of which trait came first and which came second and third? There might be a way to do it, and it's something I've been interested in, but we just haven't worked on that yet. But I don't know the answer to that question. I would suspect it, if I were to guess, it happened very early, or relatively early, because by being self-pollinating, you're able to preserve the variety, but also you're able to increase the yield, right? Because if you're self-pollinating, you're always guaranteed you're going to get seed. So that would be my guess, but I don't have any evidence for that.
Okay, thank you. The next two questions is coming from Vince Daria. So, how has domestication or evolution affected the quality in terms of energy or calorie of rice? Which has higher energy content? Is it Japonica or indica? Okay, then I'll move on to his next question afterwards.
Actually, I don't know about the calorie content. There's a different starch composition in indica and Japonica. Japonica has more amylopectin than amylose. Is that right? Yeah, lower amylose. There's lower amylose in Japonica. There's higher amylose in indica. I don't know how that translates into calories. I do know that indica has a higher yield in Japonica, at least in tropical countries. That's why it's now the preferred subspecies that's grown.
Okay. His second question is, for the DADI equation, what is the approximate time or duration used? Is it years, decades, or centuries?
It's generations.
Okay. For the gene diffusion. Okay. For the gene diffusion to take place, what does it mean by rapidly evolving species?
Right. No. It's in the diffusion equation. In evolutionary, in population genetics, when we do these calculations, it's in generations, and we assume that there's one. For rice, there's one generation per year. So that equates generations equals years. And what was the other question? Oh, rapidly evolving.
Yeah.
Oh, it's not clear what we mean by rapidly evolving. It's like, I don't know how we could define rapidly evolving. To me, it's a more operational thing. Rapidly evolving is when you can't use individual genes to trace the ancestry, because every gene has its own ancestry. I mean, there's probably a definition for it, but I just don't know what it would be.
Okay. But with climate change, has there been a significant gene diffusion change recently?
Oh, recently?
Yeah.
No, and that's one of the things that we want to do in the future, right? Why we want to reconstruct the dispersal of rice around, because what we want to do is reconstruct to see. So this is the collaboration that we're doing. I'm trying to reconstruct, using genomic data, the history of rice as it moves to Asia. Jade Guedes, my collaborator in UC San Diego, is trying to reconstruct the climates from the present into the past, including, like, there was a drought in the 19th century. We have records of that, but she's got records that go back to 1,000 to 2,000 years for precipitation, and then she can get temperature records from pollen data. And what we want to be able to do is see if we can correlate any genetic changes that have occurred in rice that can be traced to these climate events in the past. So that's the hope. I don't know how to do that. So we're going to try to figure out how to do that. And I have a PhD student now who is going to try to take up that challenge. But that's kind of the idea.
Okay, thank you. So the next question is from Raymond Tan. Are there clues in the genetic record on corresponding agricultural practices? Rice cultivation strikes me as unusually labor intensive, thus entailing a big leap from natural growth.
Yes, there are. I mean, a lot of it has to do with water management. Um, and so how extensive the water management was. Were they using rain-fed versus irrigated? When did irrigation come in? You see a lot of social organization around. A lot of social organization thousands of years ago was around water, was around how to manage irrigation. That's what created hierarchies in who controlled water. So there's that. There might, there are other things. So from that, there are other clues in the archaeological record in terms of harvest technology. So the use of sickle knives or harvest knives, for example, when did that occur? So there's some practices that you can glean from the archaeological record, and also some cooking practices actually can glean from the archaeological record. But I think you'll have to talk to an archaeologist more specifically about that. But the labor thing is actually interesting, and I actually don't know the direction of this. But there's not a lot, but there are studies archaeologically are trying to estimate the amount of labor it was required to be a farmer 5,000, 6,000, 7,000 years ago, to try to understand the labor economy and labor energy. So that's certainly something that archaeologists are interested in.
Okay, thank you. And then we have the last question. Can you comment on genomic evolution and diploidization in rice? Because during course of evolution, the duplication as well as segmental polyploidization happens. So this is from Manon Mani, also from Tamil Nadu Agricultural University.
So I mean those have occurred, but actually they've occurred fairly far back in time. These are a lot of the segmental polyploidization and duplications. Most of them occurred way before the domestication process. So people like Rod Wing actually, who are looking at comparative genomics between different species in Oryza, is probably better suited to answer that question. Because by the time you get to domesticated rice, all of those genomic evolutionary events have already occurred. These are when large segments of the genome duplicate, or even big chunks of the chromosome. But that occurred way before the time frame that we're talking about here.
Okay, we have another question from Giselle.
Sure.
We've done concatenation of few genes to generate conoidean phylotrees. How did gene integrate separate 100 gene analysis to arrive at single origins to jibe with Carlos's theoretical analysis?
So Giselle, what she used was was called BSAR. It's downloadable. It's the Bayesian estimation applied to sampling trees, and basically it feeds in you feed in the sequence, and then it estimates the probability of which tree is most probable, given the sequences you tell it to, and it will ask you to feed in 10 genes, 20 genes, or whatsoever. So concatenate, the problem arises only if you're dealing with phylogenies of species that are either very recent or very rapidly evolving. If the species are very well established, that are old species, then this is not a big issue. All the genes should give you the same picture. It's only when you're dealing with very recent species or very rapidly evolving recent species that this becomes an issue. And I can always send her the link to BEAST.
Okay, thank you. Well, I have one more question.
Sure.
So you've shown us how climate can influence how how the rice eventually evolves. So, based on your work so far, so what do you see would be the next, the future, like especially with issues on climate change and with issues on availability for
Yeah.
food?
So we, our my group especially, and if you ask public breeders, then one of the major concerns is water availability,
Mm-hmm, mm-hmm,
and how to deal with drought conditions. So that's a major concern that we're focused on. The Ziegler Family Foundation, which is a private foundation that's been funding me now for 6 years in the future, they're very interested in the question of climate change
mm-hmm.
and our work and on drought. If you ask public breeders what their major issue that they're worried about, it's drought. So that's the one thing. The temperature is also interesting because actually, especially now, even today, rice, what happens when it gets too hot is that actually the ability to pollinate and get seed actually goes down. Self-fertilization goes down. And one of the worries that's happening is that as things get hotter in the tropics, you might see a collapse in rice yields because they're no longer able to pollinate themselves. They're not able to fertilize because the temperature is too hot. Um, so, it's something that I wanted to look at, but I haven't had the opportunity to think about it. But that it's well known that hot temperatures
Mm-hmm.
reduce very hot temperatures reduce yield because they just don't fertilize the pollen doesn't fertilize the egg. So
Would you be aware of any interventions that are,
I
you
don't
know?
know. For drought, everybody's looking at drought. A lot of people are looking at drought right now. I don't think there's a good one. We were actually in collaboration with Erie. There's several genes that we're trying to test right now
Mm-hmm.
that come out of the Nature paper that I talked about earlier. We're testing right now with transgenics that we think we're hopeful. From our work on transgenics for drought, it seems that getting the plants to flower early, that's
Mm-hmm.
not a surprise. Everybody knew that. Breeders knew flowering early helps you against drought. But we are also seeing genes involved in photosynthesis
Mm-hmm.
that seem to give you higher yield under drought conditions.
Oh,
So we have two genes that are involved in photosynthesis that we're trying to make transgenics of to see if it helps under drought. So we'll see.
okay. So we have follow-up questions from Giselle. How would introduction of new rice varieties with tun-grow resistant, salt-resistant, vitamin A, CRISPR-generated varieties affect rice evolution?
Oh yeah, well right now the rice evolution is actually a misnomer because rice evolution is being done by breeders and agricultural companies. So it's no longer the natural evolutionary process. So I mean, so right now, if you can think of it, this is the next phase in evolution where we're completely directing and we're designing what traits we want in rice. So it's a completely different, and actually that's an interesting. I haven't thought about that. What is the evolutionary dynamics of that? Because now it's under complete conscious control.
Mm-hmm.
Everything I've told you in the past, only a few of it was under conscious control. But this one now, it's under full conscious control. So it's really what we make of it that is the evolutionary future of rice and other crops, for that matter.
Okay, and then one final question from Giselle. What is the prospect of aquaponics to grow specialty rice?
It's pretty good. Well, yeah, it can be good. It can be good, but it has to be under very special circumstances. Rice takes in a lot of water, so you'll really need a lot of water to grow rice. In aquaponics, I know that in Dubai and in the Middle East, they were thinking of salt tolerant aquaponic rice there. I just don't see it. The thing about rice is that it's one of those things that you want very large harvests, right? It's completely about the calories. I think aquaponics, and actually Joel can speak to this, I think should be focus more on very high value crops that, you know, are essentially are expensive in the market, because then you can use aquaponics or hydroponics or some sort of indoor farming to do it. I just don't see it for the major cereal crops, because you really need millions of tons of harvests to feed the world.
Okay, thank you very much, Michael.
Okay.
All right, before we end this session, I'd like to advertise. So let me share my screen. Okay, so next week we'll be having a talk by Dr. Angeline Lau on socio-ecological network structures from process graphs. Angie is currently an associate professor of mathematics and statistics department and assistant dean for research and advanced studies at the College of Science at De La Salle University. And during that talk, we will be having a special guest lecturate from Professor Raymond Tan on p-graph fundamentals by p-graph fundamentals. So to register, please go to bit.ly/Angie Lau. Okay, so let me check. I think we have a few more. Okay, so thank you very much, Michael. Do you have any other final words?
No, thank you very much. I hope you learned a little bit about how rice evolved, and stay tuned. I think we're going to add to this picture in the next few years as well.
Yeah, we'll look forward to those publications as well.
Okay.
Okay, thank you so much, everyone. Good morning and have a good day ahead.
Thank you, Kathleen. Thank you, Mike. Thanks, everyone.
Thank you.
Thanks. Bye.
Bye.